hopefully I can give you a little bit of an insight into what I've been working with with Reed Roberts over at NC State in this next few minutes before we all get to go have lunch. So thanks for sticking around and paying attention. These have been really great talks. So East African cichlid fishes are a really classic model of speciation. And the historical interest in these as evolutionary biologists has been, I think, pretty enthusiastic. And the lakes are called Darwin's dream pond for a reason. And that's because in the past sort of 2 to 5 million years, the Rift Valley opened up and created these massive lakes that are rivaled really only by the American Great Lakes in size. And you had this influx of riverine cichlid species depicted here, which then went and diversified across niches to evolve into what we now say, depending on if you're a lumper or a splitter, is between 500 and 1,000 of species of haplochromine cichlids that have a very diverse ecological roles. So we have these big sort of open water predators that swim around and have big teeth, and little algae eaters. And so we see really a great variety of this niche partitioning that's underlying speciation. But what I was interested in is, are there other mechanisms we can identify the genetic basis for that are reinforcing niche partitioning in this group? So this is Lake Malawi. It's really beautiful. And some of the things that we can see in the lake is all these microhabitats that have distinct environmental pressures. So here is a photograph of the rocks in the lake. And as you can see, there's a great diversity of densely packed communities filled with many species. And these have crevices and caves and places for fish to hide. The environment is really dynamic, so the waves rush against the shores and everything is constantly changing. And then we can compare that to other environments in the lake where you might see distributed communities, really fine substrate, and a completely different regime of selection pressures that would allow for different behavioral strategies to be adaptive. And so what I've been working on over the course of my PhD is identifying behaviors that may be adaptive and then trying to understand the genetic basis of those behaviors and how they relate to speciation. So lately, I've been working with Zach Johnson. He's in Todd Streelman's lab over at Georgia Tech. And it turns out that we independently were doing these sort of exploratory environment use behavioral tests, and that we were independently finding that rock species and sand species have different behavioral patterns. So throughout the course of this talk, I will indicate that the sand species are in sort of a yellow-red tone, and the rock species are in yellow-red tone, and the sand species are in sort of blue colors. And any time that a species is adapted to the rock-sand interface, it's in green, as indicated by these colors under here on my toy tree. Some features of the cichlid radiation are repeated and pervasive introgression and admixture between our lineages. So while we have an idea that there may be this initial big split between species that adapted to the rock and species that adapted to the open water sandy environments, we see reuse of adaptive alleles even between these groups. So Zach and I have been controlling for rock sand lineage in our models, but not using particular trees to control for evolutionary history. So what we find when we look across all of these many tests that I don't have time to tell you about is these distinct patterns that are associated with microhabitat use by species. And the one behavior I am going to tell you about today is this open field response. And so what we do is we take individual fish, we place them in a big white bucket, and we film them. And then we can use computer vision to get at what our behavioral metrics are. And we can look at position in the arena. We can, I think it doesn't like this real quick. Aha, okay. So we can look at position in the arena relative to the edges, and then we can also look at how fast the fish is swimming and come up with quantitative metrics of behavior. And so here we see that these are, for one particular assay, where we looked at corner position, that behavior in the arena is associated with microhabitat. So from this point on, I'm going to use summary statistics that just shows you what it looks like our overall behavior patterns by microhabitat. And what those are is that rock and intermediate species spend more time around the edges and less time in the center, and our open water species are spending more time swimming in the center of the arena. And these rocks and intermediate species are swimming more quickly around the edges of the arena. So what we have are these patterns where we have open water, middle slow swimmers, and rocky shore edge fast swimmers. And so I've taken this information and decided we're really using this as a proxy for habitat-associated behavior. And fortunately, in the lab, we have the ability to make really cool hybrid crosses. So I have a hybrid cross that's an F2 between rock and sand grandparents, and we've done QTL mapping in those, and been able to identify the complex genetic basis behind different features of these behaviors. And for the purposes of this talk, I'm just going to talk about open field swimming speed, but position in the tank and also changes over time have similar types of genetic architectures. And I'm going to focus in on this one large effect QTL on chromosome 5, which has a phenotypic effect that we would expect from our parental species. So in our cross, the F2 that inherits the allele from the rock grandparents behave more like the rock species and vice versa. So this is all good, what we'd hope to find. But unfortunately, as is the downfall of F2 crosses, our mapping interval is quite large. And so if we want to get down to mechanism and dynamics at the locus, we have to use other strategies. And so I've used we call it divergence mapping, where we bin species, genome sequence from species by the trait that we're interested in. So here I've binned them by rock habitat use and sand habitat use, and looked for regions of divergence underneath the QTL. You might notice that there are these high FST peaks across the whole chromosome, which is to be expected given that there are many, many differences between these species. So we can use the pairing of our QTL mapping and our divergence mapping to really identify the function of these high FST peaks across the genome. But this isn't a very large sample, so I took my highest FST signal from underneath my peak, and I looked at it in a lot more species. So this is a collection of data from a genome scan, from some pool-seq, and from some targeted resequencing of many species, where I now have for the allele at this high FST peak, allelic state for 39 genera and 82 species. I didn't really have enough consistency across my samples to be able to look at allele frequencies specifically. So what I did was bin everything into each species is either categorized as being fixed for one allele, fixed for the other, or segregating variation. And then I do a Fisher's exact test to ask, is allelic state at this locus associated with microhabitat? And in this case, the A allele is 60% of the species that are fixed for the A allele live in the rock versus only 15% in the sand. So we see this association between SNP genotype and microhabitat. And from this point on, I'm going to call the A allele the rock allele and the G allele the sand allele for this reason. However, that doesn't really get at the issue of potential phylogenetic signal. So I was also able to find a genus where we have all three microhabitats, but the same evolutionary history. And again, we see the same pattern, that our sand allele is fixed in the species that has independently reinvaded the sand. So, additionally, there are all the interface species, or many of the interface species are segregating the variation. So I was able to take some of these interface species that we have in the lab to then make crosses where we know the genotype at this locus for further functional validation. And just in case you were wondering, they do show the same behavioral pattern. So siblings from this interface species with the rock allele swim more close to the edges and swim more quickly than their sand allele bearing siblings. But then we can do other things like give them microhabitat choices and ask, is allelic variation at this locus associated with choosing to spend time on the rock side or the sand side? And it turns out it is. So fish that are fixed for the rock allele spend more time on the rock side. And I did do this test where I ran it where I swapped the sides. So there's that's not an artifact of my setup or anything. Well, what does it do? I've just showed you that it's associated with microhabitat and behavior, but what is this SNP actually doing? It's in upstream of 3 neural cell adhesion molecules, which are important in neuronal cell adhesion, axon guidance, and have been associated with learning and memory. And this particular polymorphism is in a conserved non-coding element that's conserved all the way to zebrafish, which is approximately 200 million years, and a very long time for non-coding region to be conserved. When I use this sequence, the other thing I want you to notice is that the derived allele is our sand allele. So it looks like the derived allele is disrupting the function of this conserved non-coding element. And when I run it through a transcription factor binding database, it turns out that our top 8 hits are known neuronal transcription factor binding sites. So I think there's pretty strong evidence that this is functional and that it's functioning as a transcription factor binding site for these genes. So I did allele-specific expression in these genes to ask whether our allele was affecting expression of any of the genes in the region, and it turns out that CHL1 is both affected and fits our pattern. So the allelic copy of CHL1 that's associated with our SAND conserved non-coding element has a twofold reduction in expression. So you break the transcription factor binding site, you reduce expression of CHL1 in these fish. Well, we're capitalizing on the fact that now this is a conserved all the way to zebrafish, and it turns out that if you knock out CHL1 in mice, they phenocopy what we would expect in our fish. So we're doing the thing that crazy people do, and we're working with zebrafish people to make CRISPR knockouts. So right now, and we have a couple of lines. We're targeting CHL1 as the gene proper, and we're also targeting the conserved non-coding element to be able to get an idea of how these are impacting behavior. And right now, we don't have stable lines, but we have looked at behavior in our injected F0s, and we see differences in how fast the CNE knockout fish are traveling, and also their startle response. So I think we have some really promising results that are, and this will be coming out shortly. So, so what we have is this evolutionary conserved regulatory element that is giving a real connection to how these fish are actually using their ecology. And we can tie that to a difference not just in ecology, but behavior. Look at the mechanism behind how these genes are impacting the brain, and also bring it out to a broader context to show that these are impacting evolutionarily conserved pathways that may be important in brain function in general. And indeed, in humans, copy number variation at CHL1 is associated with schizophrenia. So I'm hoping that what we're contributing is both something about evolution of behaviors and also filling in what we know about these genes for neuroscience in general. And for us speciation lovers, this is also a really exciting potential locus for allowing for assortative mating in the wild. So if you have the evolution of this sand allele that allows the fish to swim out further from the rocks, you've then created a behavioral mechanism for fish to separate via space. And so I think this is a really exciting potential genetic substrate for niche partitioning in this group. And with that, I have a lot of people, like lots of people who've helped me, and I really think they're all great people, so you should say hi to them if you meet them, and my funders. So thanks.